Hi everyone, very good morning, guys. I hope I'm audible and visible to all of you. And welcome to this session, for which I think I have already scared you enough in the past 2 days because the topic is such that students have sent me such scary messages as to how this gives them sleepless nights. So I'm assuming that you've not entered this class with a very positive mindset because the kind of collage and the picture in front of you certainly tells you that this is going to require an extra effort from your end to be able to concise, consolidate, and remember this for a longer time. But we'll try our best that, whatever best we can do in this table and as much as we can retain, we would do so in this class. I have sent a blank PDF, without annotations. I have sent it on the group. Right after the class gets over, I will send the annotated PDF also. So even if you're not making notes, just listening and learning, that is also good enough, right? Okay, so yes, let's get going. But for the time being, approximately 30 to 40 minutes is the duration of the class. Try to keep all the negative views that you have about these culture media, try to keep them on the side, and we will try to make it as easy as possible. Let's get going. This is about bacteriology. I suppose yes, everyone has joined in, and a quick confirmation as I can read. Yes, perfect. Chalo, let's start. We'll go in order. Cocci, positive, negative, then bacilli, negative, positive, in that order. And in the end, we'll do the miscellaneous bacteria. Perfect. Let's begin. The pattern remains the same. I've got a couple of fill in the blanks for you. So you need to answer it, and gradually we will keep removing these tapes and we'll keep unhiding the answer. So let's start with Staphylococcus aureus. Now, there is a selective media that is put up in front of you. What kind of fermentation does Staphylococcus aureus show? And remember, it shows you mannitol fermentation. So, Staphylococcus aureus is a mannitol fermenter. Now, when I know it, two properties. First, know the property, you will automatically know the culture media. It is a mannitol fermentation process, so I need mannitol in the culture media. Secondly, it will grow in the presence of salt. It is okay with salt. It doesn't have a problem with salt. So my culture media, this means, can have both. It can have mannitol also and it can have salt also. What is the name of the media? Mannitol salt agar. Now, anything else that the examiner writes with salt, like Ludlam's salt agar or any other salt agar, that also I will put under this. So I just know that anything that has salt and anything that has mannitol will come under this Staphylococcus aureus. But we're not just making a table. I'm also going to tell you one previous year question of FMG, NEET and INI CET. So now the question comes is, what color has mannitol fermentation given you? What color have you got over here? So please remember that you can very well see from the slide itself, the color that we've got is a yellow color. Why is there a yellow color? Means if mannitol was fermented, then I will get a yellow color. Means some color change is happening. I must be having some indicator over here, right? And the indicator is going to be phenol red. Excellent. Dr. Sayanthini had already answered it. The indicator is phenol red. Now, right now you've just done one fermentation, mannitol. Gradually in this class, you will do lactose fermentation also, sucrose fermentation also, so many others. So right in the end, we will make a fermentation table so that all these colors, but for the time being, remember, a trick to learn this is, no matter what gets fermented, mannitol ferment ho, sucrose fermentation happens, lactose fermentation happens, whenever fermentation will happen, the color that I will get will always be yellow. Right? That's the rule that you're taking for the next 40 minutes. Mannitol, lactose, sucrose, whatever fermentation, color will always change to yellow. So the first one is done. Staphylococcus, keep repeating with me. And also one more thing, the interesting part is, once you finish this entire table, I'm going to get you back to this slide because this slide has all the images of the entire class. So you're going to do a 30-second recall quickly of all the images in the end. So you've done the first one, the mannitol salt agar. Let's move on to the second one, Streptococcus pyogenes, which is my favorite because we know everything of pyogenes is pi. That is how you have to learn it. So how I have learned it is pi. So firstly, you look at this culture media, nothing to know. It's a reddish color culture media. So this has to be blood agar. Now they will ask you on blood agar, are you seeing narrow zone of hemolysis or wide zone of hemolysis? So I can see a lot of breakage. It's a wide zone. Number one, wide zone. Number two, it is what colony? Is it a head or a pinpoint? So it is a pinpoint colony. These are the 2 points. How are we going to learn it? So I told you na sab kuch pi y chalega. So what kind of colonies? Pine point colonies are going to be there. Number one, and wide zone of hemolysis will be noted. So pi y, that is how we are working. Pine point colonies, wide zone of hemolysis. Next question. Streptococcus pyogenes, probably a sore throat or probably some, something, a nasopharyngeal swab has been taken. How will you transport? Your lab is 5 hours away from the patient. How will you transport this? Many of you had answered it. The transport media is Pike's media. Again, you've completed. See, now you know how that pi came. Pyogenes has pi for Pike's media, has pi for pine point colonies, and has y for wide zone of hemolysis. So all your pi, y comes together. Last point, what is the selective media? For that also, I know there are too many things written over here, but selective media, very simple to learn. First, write the word Streptococcus pyogenes in front of you, and your answer is in front of you, right in front of you. The name itself is the answer. So number one, you know P. P for which media? P for the PNF media. P for the PNF media. Next, coming to the V or the Y, which I'm going to convert into a V. This is what? Very good. Most of you have answered this. This is the most important. This is the crystal violet blood agar. See, I could have just said blood agar, not regular blood agar, crystal violet blood agar. And the last one in Streptococcus pyogenes is the O. So the O is going to have this dot, dot, dot. What is this? It is granularity. Whenever you grow it in some kind of liquid media, it tends to show you granularity. Quick repeat. What do I do? Whenever I think of Streptococcus pyogenes, P for which media? PNF media. Y for what? YV, that is crystal violet blood agar. And O for what? Dot, 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 that is granularity. So pi, Y, and pyogenes. All the pyos come together. This is also done. Let's move on to another one. Streptococcus agalacti. What is this media? Very famous. They give you a lot of questions on this, and this is the famous CAMP test. How am I going to learn it? Firstly, many students ask me, ma'am, what is the full form of CAMP? You don't really have to know. I'll say it out. These are 4 scientists, okay? Christie, Atkins, Munch, Peterson. You don't have to know the name of the scientists. Together, they have called it the CAMP test. Now, remember, you are having a gala time. Gala, agalacti gala. You are having fun in where? You are having a gala time in the camp. Okay, so just before I get into the CAMP test, is there any other organism which can also be CAMP test positive? One is going to be Streptococcus agalacti because gala time in the camp. Any not reverse CAMP test. Tell me CAMP test or con positive hota hai. Even Listeria is CAMP test positive. And how am I going to learn this? Anything about Listeria that you learn, you know from my previous classes. This year's question also came on Listeria. Listeria is the drunk man. I always call Listeria as we, very famously in our rapid revision videos, we have referred to it as Tully, that is drunk man. Why do we call it drunk? Because this at some temperatures it is going to have motility, it is going to fall, and at some temperatures it is not going to fall. So yes, at some places it is going to move, at some places it will not. Just like a drunk man keeps falling, tumbling here and there, Listeria also does that tumbling here and there. Why am I teaching you that over here? Because of now when someone has gone camping, so camping is some place he would also want to drink and enjoy, right? So how will he have a gala time in the camp? Of course, by drinking. So this is just a way of learning that to have a gala time in the camp, a person would be tumbling and a person would be drinking. So these are the 3 things that you have to put together. Very good. Everyone knew this. Some of you answered Clostridium perfringens. That I'll teach you in 20 minutes because that is reverse CAMP test. वो उल्टा है. Abhi के लिए what is this CAMP test? Please do the labelings also for me. You can see there is one line over here and there is one line over here. So Please remember this vertical line that you have is Staphylococcus aureus, and this line, horizontal line, is Streptococcus agalacti. So this, always remember, this line is going to be agalacti. Gala time in the camp. What will you do in the camp? You will lie down, है ना? So gala time is lying down. So this lying down line is going to always be the gala line. This is gonna be the agalacti. The gala line is always lying down. And can you see what has happened? One line is like this, one line is like this. What do you see at their junction? At their junction, I can definitely notice enhanced hemolysis. That is going to be the camp test. Everyone, quick revision about camp test. Camp name comes from the scientists. Number one, camp test. Camp is having a gala time. So agalacti, gala time by drinking and tumbling. So even listeria shows you. What are the labelings? The vertical labeling is of Staphylococcus aureus, and this horizontal one is gala time is lying down and relaxing. So that is Streptococcus agalacti. Everyone's okay with this? Can we move on to the next slide now? Chalo, another streptococcus, that is Streptococcus pneumonia. This has a very classical picture in front of you. It's written in front of you. I have nothing more to show you. Can you see these colonies which are elevated and then in the center they are depressed? Are they reminding you of carom, that coins that we play? Yes, so carom coin colonies, or also known as Draughtsman colonies. Why Draughtsman? Because center is depressed. The periphery is like this and the center is depressed. So carom coin colonies or Draughtsman colonies are seen in Streptococcus pneumonia. There is no mnemonic needed for it because it's very, very evident and everyone knows this. So there's nothing more that I have to teach you out here. Let's go to Enterococcus. Enterococcus may always remember the E that you will make for Enterococcus will always be converted into a B. Why? Because whether you look at the shape of Enterococcus or the ability to bile ability or the media, everything comes with B. So remember what you're going to do. Enterococcus, the E is going to change into a B. So that means B. What is B looking? Is this looking like spectacles? Yes. Is this looking like spectacles? So the B shape will tell you that this is spectacle shaped organism. It has a spectacle shape, number one. Next, B will tell you bile से relation है. So remember the culture media that you have is the bile esculin azide agar. Even if you can't learn the full name, learn something with B. It is bile wala agar. And can you tell me the color? What is this color that you have? Again with the B. The color that I see over here is a black color positivity. So will you learn Enterococcus is everything B, B, B. Whether I look at the shape, that is spectacle, or I look at the B for bile ability, or I look at the color that comes, everything comes as black. Okay, let's move forward. Dr. Vishwas has said 6.5% bile soluble. I think thoda sa you have done, you've revised it. I'm definitive that is why you come up with that color, but 6.5% of Enterococcus was NaCl. Bile is 40%. You've just confused out on those numbers over there. So just a quick revision needed. Okay, let's come to the next one. Neisseria, we know two types, meningitis and gonorrhea. So two things that they ask you over here. Neisseria sample has to be transported and it has to be grown. What are the culture media that you have? Let's start one by one. If it has to be transported, you are using the Ames or the Stewart's media. Ames or Stewart's media, what does it have in common? Because they contain charcoal. The Ames or the Stewart's media, they are charcoal containing media and they are the ones that are used for Neisseria, both meningitis also and gonorrhea also. I'll write both over here. Whereas the real question that comes is on this, the modified Thayer-Martin medium or the New York City medium. This question which I'm going to highlight, modified Thayer-Martin medium, is very, very important. Some of you might have seen INI-CET students. This one is for you. You might have seen they've asked you what is the modification in the Thayer-Martin medium. What extra has been added? Why am I not calling it Thayer-Martin medium? What is modified Thayer-Martin medium? So remember the modification. Anyone? What has been added? Something known as trimethoprim has been added to the culture media to be able to make it modified. It might not be visible behind me, so I'll just erase that. Yes, it is modified, meaning that it is trimethoprim that has been added. ठीक है, चलो, everyone's okay with this also. I hope things are going well up till now, so that we can now move on to the next one. And here I am back as well. Okay, so coming to the next, this is one of my favorites, Corynebacterium diphtheria. This is one of examiner's favorite also. Corynebacterium diphtheria, diphtheria, that bull's neck and pseudomembrane, that famous picture that always comes. They ask you two things: enriched media, culture media. So I think you remember this mnemonic by now. Enriched. How many alphabets of enriched? We have 8 alphabets, and this definitely puts a smile on my face, because recently I was seeing some students had made memes and reels also out of this, counting the alphabets and making the letters. So enriched media has 8 alphabets, and Corynebacterium diphtheria, iska dusra naam, other name of Corynebacterium diphtheria is Kleb Loeffler bacillus. Kleb Loeffler bacillus. Why? Because Loeffler was the scientist. So everything about Coryne will come out as Loeffler. And Loeffler has how many alphabets? 8 alphabets. So if I ask you, what is the enriched media that you have for Corynebacteria, you will say, ma'am, 1, 2, 3, 4, 5, 6, 7, 8. Loeffler serum slope. And how much time does it take? 8. Everything has to be 8, so it's going to take 6 to 8 hours. Easy. Repeating once again, enriched has a total of 8 alphabets. Loeffler has 8 alphabets. 8 hours is the time taken for it to grow over here. Whereas what is the selective media? Selective, can you see a blackish color media over here? Selective media is the potassium tellurite agar, PTA, or the Tinsdale media is also used. Potassium tellurite. How do I learn this? Firstly, you will say that, ma'am, enriched was taking 8 hours to grow. Potassium tellurite is selective. Selective things take more time, right? So this is going to take 48 hours to grow. This is going to take 48 hours. So now tell me a question from previous years. If you want to do the diagnosis, listen to me really carefully. If you want to do the diagnosis of Corynebacterium diphtheria, what are you going to use for the early diagnosis? Which culture media? If you want to diagnose it early, I will use Loeffler serum slope. But which is the best culture media that you will use? Potassium tellurite agar. Kuch common dikha? Why did I call it? If you want, I'll write it as a full form. Why did I call it potassium tellurite agar? Because whenever I use the word tellurite, I can see the color in front of me. The color is gonna turn black. Tellurium is a metal, hamesha black hota hai. So that's how I learned that, okay, whenever the word tellurium will come, I will see a black color in front of you. Tell me one organism where I just now told you bile esculin azide agar, and that was also coming out as black in color. Quick revision, we need to keep revising. Bile wala, which was coming as black in color, which organism was that? That was Enterococcus. This is another black one that I'm telling you, potassium tellurite agar, because tellurium also turns black in color. So Corynebacterium is done. Now let's come to, can you see the longest collage is over here? There are so many pictures that I have in front of you, because I'm talking about next a bioterrorism agent, Bacillus anthracis. So obviously the most deadly and the maximum number of things are going to come for Bacillus anthracis. So let's get going. Some of them are for FMG and NEET. Some extra points I have got for INI CET students also. So first one, I've got all of them hidden. As you'll keep answering, I'll unhide the answer. What is this? What is this appearance of hair known as? So this is the Medusa head appearance. Very good. So this, I think everyone knows, it shows you the Medusa head appearance. After that, what are the things you don't know probably are the next three. If you start looking at these three I usually don't teach, but they are important from INI CET point of view, and I'll give you a mnemonic also after that. Can you see that if this one, does it have that like glass pay during winters, that frosting comes? Same way, this is a frosted glass appearance. It's looking a little frosted out here. Look at the next one. Next appearance, it has a tail. It has a tail. So comet tail. You can see another tail over here, another tail over here, another tail over here. Comet tail appearance. Coming to the next. Everyone must have sometime or the other, you must be beating eggs. When you beat eggs, this kind of appearance comes, beaten egg appearance. So these three are important for INICET. Bacillus anthracis shows you the frosted glass appearance, the comet tail appearance, and the beaten egg appearance. This is extra information. Now let's come to this one. This is, especially the girls should be able to identify this, because this is looking like that beautiful pearl string that we wear. That pearl string of white pearls, beautifully arranged. What is this? So simple, it's a pearl string appearance, string of pearls, but which agar? This is not just blood agar. So please note over here, this is going to be penicillin agar, which shows you string of pearls appearance, PEPE. If someone asks you a single question, simple wala question, how do you see the string of pearls appearance? When I will be adding the penicillin agar, as easy as that. PE for penicillin, PE for pearls. You don't see it on a regular blood agar. Finally, one of examiner's favorite. What is this on the last part that is written? Gelatin stab. I'll enlarge it. Yes. What is this showing you guys? What is this appearance known as? Why is this going like an ulta tree? Because that is the name given to it. It is the inverted fir tree appearance. In some time I will come to inverted, na ulta tree. It is coming as an upside down tree. So inverted fir tree appearance is gonna be noted. So how do I learn all of this? Firstly, Bacillus anthracis. Is it sounding very nice and happy bacteria, or is it like a very deadly bacteria? So of course it is a very deadly bacteria that you have. So what you're gonna imagine with me is a very deadly looking ghost, right? So if you want, although my drawings are not at all nice, but if you want, we can draw one. So you're gonna imagine with me a deadly ghost. And how are the hair of the ghost? If I always ask you कि ghost का hair कैसा होता है, so you will always tell me that the hair are gonna be like a Medusa. This is how the hair of a ghost are gonna be. So this is the story of Bacillus anthracis that you have, right? So you have this, and we are actually going to draw a very scary one, because I can see how I'm drawing is coming out as very scary. So number one, we can see a deadly ghost because she is going to have Medusa head appearance. Next, what are they going to wear? If you see the ghosts, they usually tend to wear a lot of beads, and the ones who do black magic, they tend to wear a lot of beads and a lot of ornaments, which are a little scary for us. So we will call this the string of pearls appearance. Everything about Bacillus anthracis has to be deadly. Why? Because bioterrorism agent hai. Bioterrorism category A. So everything has to be deadly. Okay, next. Whenever you see in Bollywood movies especially, where is the ghost always present? Under some tree, right? Some tree, or hanging on some tree, or lives under the tree. So there is always a reference of a tree that will be made when they are talking about a ghost. So yes, I can see over here, this is the inverted fir tree appearance that you have, right? So these are the three most important things. Baki sab you can learn or not learn, like frosted glass, comet tail, and beaten egg appearance. All of that is okay if you can learn, but these three you have to. So remember, the deadly Bacillus anthracis has the deadly hair, is always living under the tree, and is always wearing those kind of scary ornaments. So string of pearls appearance. Okay, O Stree Kalana. So yeah, for all those who watched Stree, this is somewhat like the appearance of Stree in the Bollywood movie. Exactly. But in before I forget, all of this is said and done. What is the selective medium that you have? So the selective media is something different, and that is the PLET media, right? This diagram wasn't in the main video, Dr. Hiren. Yes, because this is a mnemonic that I made last night. So this is the most recent mnemonic that I have added in our microbiology pool of mnemonics. Okay, chalo, coming back, selective media is what? Selective media is the PLET media. That is something which is extra. Okay, now coming to the next favorite of mine, Bacillus cereus. Bacillus cereus, what does it cause? Firstly, this I think everyone knows, C for cereus, C for Chinese food poisoning. Whenever someone consumes Chinese fried rice, so Chinese wala food poisoning is going to be Bacillus cereus. And now, it is not nice to name out politicians on a public platform, but this is something which I'll give you today. I'll give you every day. I give you homework which is academic. Today I'll give you non-academic homework. Do go and Google out or watch it on YouTube. A very serious Bacillus cereus, a very serious politician, in the middle of her speech started speaking words like you must have seen rumba rumba humba humba. You must have seen that that speech of hers, and no one understood the meaning of the words. So if you look at the first alphabet, that's the name of that particular politician also, right? So remember it, she's a very serious politician. That is her name, whom I will not name. And then the rumba rumba reminds you of Maipa Pemba. So if you don't know what I'm talking about, then probably you've been studying too much and you don't have an idea about what is happening since 2-3 years in the outside world. So just Google it out. Even if you write rumba rumba, so you will get to know what is this Maipa Pemba. Okay, so serious politician, what did she end up saying? She ended up saying, okay, Dr. Farman, yes, some of you had tagged me in that meme also. Every time I tell you that this is the mnemonic that should stay only amongst us, I have always seen that next day that is amongst us and the entire Instagram world. So that's something which I've noted with students. So yes, this is Maipa Pemba, reminding you of Rumba Rumba with serious politician. Okay, this is also settled, and this photo I think of Bacillus anthracis you will never forget. So now let's go further. Bacillus are done. Now we need to come to all the Clostridiums in one slide. Let's start. Clostridium tetani, and the firstly, whenever I say any Clostridium, tetani, perfringens, difficile, kuch common hai, something common amongst it that you have, these are all anaerobic organisms. These are all anaerobic organisms that you have. They don't need oxygen. You don't have to give them oxygen. They are going to survive without oxygen. So whenever I have to go for an anaerobic media, what is the media? Robertson's cooked meat broth, means I will use Robertson's cooked meat broth for Clostridium tetanus also. I will use Robertson's cooked meat broth for Clostridium perfringens also. But I see a change in the color which I want to tell you, which you must have heard. Clostridium perfringens, when they say on Robertson's cooked meat broth, you must have read that it gives you something called saccharolytic reaction. And many students ask me, ma'am, color kya aega? Saccharolytic means pink color. Whereas Clostridium tetani on Robertson's cooked meat broth gives you a proteolytic reaction, and that is going to give you a black color. So now how do I learn what is saccharolytic, what is proteolytic, what is pink, what is black? So काफी difficult हो जाता है to learn that. So if you've gone through rapid revision videos, main videos, or whichever platform, you must have seen that there is one very famous mnemonic. When I cannot decide on what mnemonic to make, I make a mnemonic on my own name, and I feel that that way you will remember me in the exam also. So just in case you forget in the paper, कि perfringens में क्या था? So what is my name? What a question to ask. So whenever you see, okay, the perfringens का P has come, so अब ma'am का next alphabet has to come. So what is the reaction that it is going to show you? It is going to show you saccharolytic reaction. And ma'am का favorite color, that is going to be pink. So always remember, perfringens shows you a saccharolytic reaction which is going to be pink in color, and then tetani becomes opposite. Tetani is going to be proteolytic with black in color. Okay, so Preeti Sharma pink, and you will realize this in fungal chapter also and a lot of other chapters also, that many mnemonics I make on my own name, so that you can learn that okay, this subject you studied from this teacher, so her name should help you in some way at least. Okay, this part is done. Now coming to the next. What is this? Clostridium tetani is showing you this round kind of growth. What is this known as? This is the swarming ability. We call this swarming. ये जो round जा रहा है, that is known as swarming ability, and swarming is famously shown by some other bacteria also. Jaldi se batao. Swarming is shown by PVCs. So actually I tricked you guys that this is only a culture media class, but I've extended it into a mini or a micro bacteria revision also. Swarming, ये जो round है, that is shown by, of course you just wrote Clostridium tetani, but other than that it is also shown by P for Proteus. It is shown by V. कौन सा Vibrio? Vibrio cholera? No. Look at the previous one. VP, Vibrio parahemolyticus. Vibrio parahemolyticus. And S for the last one, that is Serratia. There are a lot of organisms. Honestly, if you read textbooks, there are 10 more organisms that can show you swarming. We have to learn the most important, and PVCs is how you are going to learn it. So Clostridium tetani is done. Now let's come to Clostridium perfringens, and I think the famous South hero that everyone is of course knows, Senior NTR. That is how you are going to learn Clostridium perfringens. So what is Senior NTR? The pictures are given in front of you. Quickly highlight karo and finish it off. Okay, what is senior? What is this S over here? I can see some, some storm forming, some bubbles forming, kuch aisa bubbly and volcano eruption type of thing. So something stormy is forming over here. S for stormy clot formation. S for stormy clot formation, number one. N for what? What is this plate known as? N for Nagler's reaction. Of course, I am not going into the pathogenesis of each one of these because then the class will be for 3 hours or something. That will take on gradually with more sessions. Abhi ke liye we are enumerating the culture media. So N for Nagler's reaction. T for what? I can see these target type of hemolysis. Double hemolysis. So T is going to be target hemolysis. And the last one, senior NTR. This is looking similar. Sometime back we did something called the camp test. Remember, I told you camp was gala time in the camp, so listeria and agalacti. Now I am adding something known as reverse camp test. What is reverse camp test? You'll ask me what is vertical, what is horizontal once again. So now I'll ask you, who was always going to lie down and have a gala time? That is always agalacti. So over here also, Streptococcus agalacti is going to have a gala time, is going to lie down and relax. The person who is standing has changed. And this time the person who is standing is Clostridium perfringens. That is what is reverse camp test. Let me repeat. Sometime back we did camp test. In camp test, who was standing? In camp test, Staphylococcus aureus was standing. Whereas in reverse camp test, as you will notice, it is the Clostridium perfringens which is standing. So I realize the standing person changes. The lying down person is always the gala relaxing person, that is Streptococcus agalacti. Is this okay? Dr. Urvashi, you are right. For Nagler's reaction, we use the egg yolk agar. Perfect recall on that. Excellent. So Clostridium perfringens is senior NTR is settled. Clostridium difficile, when you pronounce a lot of things become simple. Difficile, if you see, it has that typical CC wala sound. So the culture media are also gonna be CCFA, CCYA. You can go into the full forms of these, but again, examiner doesn't ask, so it's not important. CCFA, CCYA are for difficile. Okay, in a while I will also come back to this and revise. Abhi ke liye this seems to be done and we can move forward. Remember, I was talking about a lot of fermentations. Sometime back we spoke about mannitol fermentation. Now that time has come that we talk about the next one, and that is lactose fermentation. Before that, before I get into the culture media, it's your revision time. Can you tell me what are the lactose fermenters? See, I have given you a big hint. All the KKK sounding ones are going to be lactose fermenters. That means E. coli, Klebsiella, E. coli, Klebsiella, and everything that is Enterobacter, Citrobacter. All these things which have the K sound in them, they are going to come in lactose fermenters. Repeating once again, and this is particularly important for INI-CET students. E. coli, Klebsiella, Enterobacter, Citrobacter, these are all gonna be the K ones, that is lactose fermenters. Means everything else is non-lactose fermenters. Now if I ask you what is Salmonella, what is Shigella, what is Proteus, what is Yersinia, you will say, okay ma'am, now do you hear any K sound in them? Salmonella, Shigella, Proteus, Yersinia, no K sound, no lactose fermentation. So non-lactose fermenters. But the trick, late lactose fermenter. Late lactose fermenter is Shigella soni. Late lactose fermenter is Shigella soni, and I think we know this mnemonic which is age old, that is Soni slept till late, sota raha till late. So Shigella soni shows you late lactose fermentation. This is also settled. Your theory is settled over here. How did you test for lactose fermentation? Are these two culture media. What can you see? One is McConkey and one is Cled, and they also have that KK sound in it. They also have McConkey and Cled. So you will know that, okay, they are all for that lactose fermentation. But now you need to tell me with Cled, what color do you see? I always told you, fermentation hoga to what color? Fermentation hoga to you are gonna get a yellow color. That was the rule. When mannitol got fermented, it was a yellow color. Lactose gets fermented, it's a yellow color. But the indicator will change now. The indicator over here is B. Can't you see a blue color in the background? Ma'am, background to is culture media ka blue lag raha hai. Bromothymol blue. BTB stands for bromothymol blue. So the indicator has changed, but it will ultimately change to yellow color, except for, I told you everywhere it is yellow, yellow, yellow. Only exception is this McConkey. McConkey is the only place where the fermentation that happens gives a pink color. Can you see? The fermentation that you have is gonna give a pink color, and the indicator over here, who's gonna tell me this previous year question? The indicator over here is neutral red. Ma'am, kaise yaad karenge? Something that's coming to your mind which I have told you. I have told you that McConkey is a pretty pink plant. McConkey is a pretty pink plant. What is pretty pink plant? Pink I have understood. Acha, the color that I'm getting is pink. But what is plant? The composition. Very famous previous year question. Please mark it over here. Pretty pink plant tells me P for it contains what? Everyone. Quick fill in the blanks. P for peptone. L for, of course, you are testing for lactose. So lactose you will have to add. It is an agar. Agar, common sense, you will have to add. The indicator, you will have to add N for neutral red. That's how you will never forget. And finally, T for taurocolate. Taurocolate might sound very fancy. Bile salts, no? Sodium taurocolate and all of that. So simple, they are nothing but bile salts. Khatam. So now this is how I'll always remember. All the fermentations, mannitol fermentation, sucrose fermentation, lactose fermentation, sab yellow, yellow, yellow hoga. But McConkey is a pink color pretty plant. Pink pretty plant because this gives you a pink color on fermentation and the composition is plant. Very good, Dr. Urvashi. Better kaun sa hai in dono mein se? Cled is considered better than McConkey nowadays. Perfect on that. So this much is done. I want to ask INI CET students a question. Even NEET PG and FMG ke liye bhi important hai. Tell me, if I have to differentiate between E. coli and Klebsiella, are both of them lactose fermenters? E. coli, K sound, Klebsiella, K sound. Are they lactose fermenters? Yes. Will they both give a pink color? Yes. So now look at the next slide. This was a McConkey agar and I could see that there is a pink color that has come. Is this E. coli or is this Klebsiella? How will you differentiate? I am saying the answer to this is Klebsiella. How did I get to know that ये E. coli nahi hai? This is Klebsiella. This is not E. coli. How did I get to know this? Because I could see that I am able to, like a string, I am able to pull it up. It is stringy. Why is it stringy? Because anything that has a capsule, will get stretchy and stringy like this. E. coli does not have a capsule. It's not gonna be stretchy and stringy. So if you have a McConkey and a pink color and they ask you E. coli hai ya Klebsiella hai, you are gonna see for the string. If the string is there, means there's a capsule. Capsule hai, matlab it is Klebsiella. Very good. All of you have given a correct answer. So this is also settled. I want to come back. This category is done. What is this category? Salmonella, Shigella, Proteus, Yersinia. Especially this was the recent FMG question also. This was the recent NEET PG question also. Salmonella and Shigella. And I have always called them Salmonella, Shigella sisters. Because everything, देखो, clinically तो अलग है. Totally different. जल्दी से बताओ, what are they different in clinically? What does Salmonella cause? Salmonella causes typhoid, common sense. Shigella causes dysentery. So they are clinically very, very different, but all their culture media are exactly the same. So that's the best part. I just have their sisters, now they are always going to stay together. All their culture medias are the same. So let's learn them. Before I start, what does enrichment mean to you? What is the meaning of the word enrichment? Enrichment, not enriched. enrichment means something is gonna turn liquidy. Something is gonna turn liquidy. So I will always use the word broth. Broth will mean like chicken soup broth or any broth that you have. Broth is always liquid, watery, right? So enrichment जो होता है, that is liquidy. That is why you will always see the word broth written. So selenite F broth, tetrathionate broth, are both of them used for Salmonella and Shigella? Yes. दोनों के लिए same enrichment media. Talking about selective media, lots of selective media, but I see something common. देखो, एक तो है Salmonella Shigella media. How easy to learn, very simple. One is enteric agar. Enteric is what? Typhoid is nothing but enteric fever. So common sense, the enteric agar will be for enteric fever. So ये दोनों तो common sense से निकल गया. Next is going to be XLD and DCA. So we realize that the deoxycholate agars are going to be for Salmonella Shigella. So you will say, ma'am, their selective media are also the same. Whether you talk about the deoxycholate ones or the Salmonella Shigella or the enteric agar, both of them will grow on all of these. How do I differentiate? Now it's time that you create a fight between them. There is a medium which is known as the Wilson and Blair medium. There is a medium known as the Wilson and Blair medium. So how I learned it is that Wilson and Blair medium, will it be able to grow Salmonella typhi? Yes, it will grow Salmonella typhi. Will it be able to grow Shigella? No, it will not. So who has finally created a fight? A boy has come and created a fight between the these two sisters. Otherwise they were always together. Dekho, enrichment mein together, selective mein together, but finally there's a boy called Wilson, and over them these two sisters are fighting. And Wilson, if I write Wilson, you will say, okay, Wilson is the name of the boy who's created a fight. Wilson and Blair. Wilson and Blair, what color has it given? This is the FMG question that came this year. It has given a black color, because obviously, has he done a good thing by creating a fight between two sisters? No, he's done a bad thing. He's done a bad thing, so he should be blacklisted. Okay, so finally tell me, Wilson has won which sister? Wilson is finally gone with which sister? Salmonella sister. So trophy mila hai, Wilson को एक trophy मिला है. He's won. So which trophy has he got? Has he got Salmonella typhi वाला trophy or Shigella वाला trophy? So he's got a Salmonella typhi का trophy. So remember, Wilson gets typhi का trophy. Okay, that is what he has won. That is what he has won. He goes with typhi, and that's a bad thing to do. He should be blacklisted because he's created a fight between two sisters. So the color that you have is black. Random stories we have to come up with. Someday I'll become more of a storyteller than an educator, I feel, because micro has no other way other than these random stories that you can come up with to learn it. Okay, this is also done. Now I can move on to the next one. So many. So now this entire fermentation story is settled now. Everyone's okay with this fermentation story. Now I can go on to another fermentation that you have. What is this? But ek hi bacha, sucrose fermentation. You say, ma'am, I know any fermentation you say, the color is always gonna be yellow. Sucrose fermentation is for Vibrio cholera, the rice water stools. So I'll, without asking you, I will write over here. Sucrose fermentation is gonna give a yellow color. Okay, what is this culture media known as? This is the Dr. SS had asked, selective media do we need to learn? Dr. SS, your name is SS. You are a part of Salmonella Shigella family because your name says that. You have to learn the selective media. You especially have to learn the selective media, and everyone else also has to learn. Okay, coming back, how do I test for sucrose fermentation? Sucrose fermentation is going to be Vibrio. So TCBS का full form. Thiosulfate citrate bile salt sucrose. नहीं भी याद रहा तो at least remember S is gonna be for sucrose. So I've got a yellow color. Vibrio is sucrose fermenter. But what is the indicator over here? Over here also it is bromothymol blue. So अब थोड़ा सा confusion होगा. Colors तो sorted है. Whatever is there, color is always yellow except for pink plant of McConkey. But indicators में थोड़ा confusion है. That I'll make a table for you in the end. Which media has which indicator. Okay. So Vibrio is done. I can see this. And which Vibrio am I talking about? I am talking about Vibrio cholera. Vibrio cholera is a sucrose fermenter, not other Vibrios. Okay, coming to the next, Pseudomonas. Actually, this doesn't have any culture media, but the color of Pseudomonas is so important in the exam that I had to show you this color. What is this classical blue-green color known as, guys? Just tell me the name of the pigment that you need to know. It is pyocyanin. Pyocyanin is very important when you have for Pseudomonas. Meanwhile, Dr. Vishwas, you are right. If you guys want to know the transport media for Vibrio, V for V, Vibrio ka transport media, I'll add that since you mentioned it. Transport media is Venkatraman Ramakrishnan medium. In fact, there is one more. Carrie Blair medium is also there. Let me take a test. 3 transport medias quickly. I know I've been writing. Now it's your time. And 45 minutes are up, I know, but I promise last 15 minutes mein the session will be done. Okay, first thing, tell me the transport media that you had for Streptococcus pyogenes. Jaldi batao, we have only 30 seconds for this. Streptococcus pyogenes, transport media, pi you had learned, so the transport is Pike's media. What is the transport media for Neisseria, meningitis and gonorrhea? So that is the Ames or the Stewart's media. And third, what is for Vibrio? So VR media and Carrie Blair media. These transport medias you have done up till now. Okay, now Pseudomonas ka not a culture media. I just wanted to revise the name of this beautiful blue-green pigment for Pseudomonas. That is done. Okay, let's go for the next one, and that is known as Burkholderia. The name of the media is the Ashdown media, but you need to tell me what is that disease. Burkholderia ki wajah se, what is that disease which is caused? Because if you know the name of the disease, then the culture media is mnemonic apne aap banega. Yes. Have we heard of something called this? Something which was in soldiers, something associated with a time bomb. Vietnam time bomb, kuch suna hai? Perfect. So Vietnam time bomb was a disease. A time bomb, a bomb will put everything down to ashes, and ashes means that the name of the culture media is going to be the Ashdown media, is going to be the Ashdown media. Okay with everyone? Yes, can we move forward again? Next wala to what to teach you living in India. If you don't know the culture media for Mycobacterium TB, then you're probably not living in India. It is the Lowenstein Jensen medium, requires no mnemonic. What actually they will ask you is what over here is this: what kind of LJ media colonies are they? So TB shows you rough, buff, tough colonies. That is how you learn it. It's a rhyming thing that you have. You have rough, buff, tough. Bas itna yaad rakhna hai. Baki LJ media, I don't want to elaborate further. It is understood. You all know it. What is Mycobacteria leprae? I have, for everything I made a culture media. Here what I have done is I have made an animal, because this is non-cultivable. You cannot grow it on a culture media. You have to grow it with animals in animal studies. Tell me something, what animal is this? Even if you've forgotten, maybe you can count. It has 9 bands. It has 9 bands. This is the 9-banded armadillo. This is the 9-banded armadillo, which tells me that for Mycobacterium leprae, you will have to use an animal inoculation and not a culture media. So, of course, this is also settled. Now we have only 2 more slides left. We've gone into all miscellaneous wala organisms. Okay, let's start. What is this one? Haemophilus influenza. I have always told you the other name. Haemophilus, F sound, influenza, F sound, known as what bacillus? Fever bacillus. Fever bacillus. Why am I focusing so much on Haemophilus influenza fever? Itna FF sound kyun aa raha hai? Because it needs 2 factors. It needs factor 5 and it needs factor 10. Lot of FF factors it needs, and this concept is known as satellitism. This is known as satellitism. So if I go zooming into it, I will realize that over here they have put a line of Staphylococcus aureus. And Haemophilus influenza kahan hai? Haemophilus influenza is just around it. So for example, if this is Staphylococcus aureus, Haemophilus influenza is going to be like a satellite around it. Satellite ki tarah roam karta hai. So satellitism, that is the phenomena shown by Haemophilus influenza fever, FFF. That means we are talking about factor 5, factor 10. Okay, Haemophilus ducrei. Before that, you will tell me Ducrey causes a genital ulcer. This much I'll tell you. But which genital ulcer? Painful or painless? I think this we all know that ma'am Ducrey has crying. The patient will be crying with pain. So it has a painful genital ulcer, right? So now if someone is crying in pain, what is the first food item that you will give to that person? Kya he or she is crying. Let me try and give him some, maybe a chocolate, right? So crying person, because he's crying with pain, I would want to give this person a chocolate agar. Another random story that we have to make. So person is crying too much, I'm gonna give that person chocolate, and alone chocolate will not help. You know, he's cried so much, you have to give him little bit of extra energy also. To that chocolate, I will add some multivitamins. I will add Isovitalix. So this is a question that they ask you, that do you use plain blood agar for the growth of Haemophilus ducrei? No, this person is crying with pain, उसे कुछ energy चाहिए, sweetness chahiye, so I'm going to give that person chocolate, and I'm going to add some growth factors to it, some multivitamins, some Isovitalix or fetal calf serum. This will give him some kind of chocolate for happiness and Isovitalix for energy. Okay, next, this is my favorite, Bordetella pertussis, because whooping cough hota hai isse. Pertussis matlab whooping cough. This is the easiest one to learn, because look at the culture media, guys. Bordet gengu media. How easy. Bordetella Bordet gengu media. Do you think they're going to ask you this? Like, this is common sense. Bordetella Bordet gengu media. They will ask you the appearance. So when a person has whooping cough, how do you think you will take the sample? This, for example, is the culture plate. This is the Bordet gengu culture plate. You will place it in front of him. You will say cough on this. So the person will cough. All the droplets will get deposited. All the droplets will get deposited here. See, this comes exactly as droplets. Can you see the colonies over here are coming exactly in the form of droplets? Bisected pearl appearance. Dot 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 colonies banega. So bisected pearl appearance, and imagine if all these pearls end up fusing. All these pearls end up fusing, it will look like aluminium paint. So very simple to learn. Bordetella, Bordet gengu media, whooping cough. So person is coughing, secretions. Would dot 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 secretions will remind you of bisected pearls. If they fuse, it will be aluminium paint. Coming to the next one, brucella. Brucella is something that can happen in actually more than humans. You will study it in animals only. So humans में भी होता है, और it comes from the animals na, so it's happening in animals also, it is happening in humans also, so it is happening in both places, animals and humans. So I will need a culture media which has two things, सब कुछ two चलेगा for brucella. So it happens in animals and it happens in humans. The culture media that I have is the Castaneda biphasic media. देखो, what is biphasic? Two things it has. It has something which is solid and it has something which is liquid. This is the solid part of the media. This is the liquid part of the media. So simple that brucella is going to happen in animals also, humans also. You will have a solid culture media also. You will have a liquid culture media also. Both are there. So I call it biphasic media. But now I don't know why we want to learn this. But if you have to do the diagnosis in animals, what will you do? I will take what secretion from the animal? I will take the milk from the animal. I'm going to take milk from the animal and I'm going to do the milk ring test. Can you see these white color samples? These are all milk samples. And if you can see, a ring has formed on top of them. So animal se to milk hi aayega, right? So this is a special animal testing that is done. Milk ring testing is what you have to know. Finally, coming towards, okay, yes, Rose Bengal test is also there. Milk ring is there. Very good, Dr. Shweta. But image comes of the milk ring test. Okay, the last slide that you have. Leptospira. If you've seen the notes, I always write it as leptospira. What is this R that I write over here? Because remember, leptospira occurs in rainy weather, number one. This occurs when rat urine contamination occurs. So rat urine goes into the rainy water. So rainy water will be there. Rat urine will go into the rainy water, and which field has a lot of water? Rain water is needed for the growth of which? So rice workers, rice field, paddy field workers, they are going to be affected. So if you get a history of rat urine contaminating rainy water and going into the rice fields, you are dealing with leptospira. And can you see In these, the last R, this is one test tube, this is another test tube, but in the second test tube I see an extra ring over here. Extra ring is formed, and that is very good. The last R, that is Dinger's ring. That is Dinger's ring, and this is on EMJH media. ये थोड़ा सा tough है, EMJH media. This is the ratta thing that you have to do. So I'll repeat, guys. We have rainy water, rat urine, rice field workers, and a ring that is formed, and that is Leptospira. That is how you're going to learn it. R. Okay, coming to Legionella. For this, I didn't give you any mnemonic. This is BC buffered charcoal yeast extract. Full form नहीं जानना. BCYE media, but यहाँ पर ना confusion starts. There are a couple of short forms that we've done. Number one short form is, let's see if you can revise them. We did something called Mypa Pemba. Mypa Pemba was which organism? This is one. Then we did CCFA, CCYA, and now we are doing BCYE. These three set of short forms will trouble you. So सबसे पहले Mypa Pemba, that rumba serious wala politician, Bacillus cereus. This I will never ever forget. CCFA, CCYA, this was the organism that had a lot of CC in it. Difficile, Clostridium difficile. So this is also settled. So now what I'm left with, BCYE, that is going to be Legionella. Now there are no short forms that are going to trouble me. So iske liye mnemonic nahi chahiye. For the other two you have a mnemonic. What is left goes under Legionella. The last one I think is the easiest, simplest of all. We've done this yesterday also. What is this colony, guys? What is this colony known as? Classical one, Mycoplasma ka colony, that is known as the fried egg appearance. This, I don't think I want to wait for your answer. This is the fried egg appearance, but I want to ask you something. Do you know this fried egg and Mycoplasma is very, very chota. It's the smallest bacteria that you have. Right now you're seeing it. Actually, you can't see it with your eyes. You need a microscope. It is that chota. So how will I see it? I put a stain on top of it, which is known as the Dines stain. This is again important for INI-CET. Wherever I've written INI-CET, na, the FMG students can ignore that. That is particularly for INI and NEET PG students. Okay? Chalo, Mycoplasma, fried egg, to everyone knew, but they are very chota eggs. So I need to add some color to be able to see it, and that is going to be the Dines stain. Last question for you guys. What is the other name of Mycoplasma? It is PPLO bacteria. The other name of Mycoplasma is PPLO. If you want to know the full form, pleuropneumonia like organism. PPLO full form है इसका, but no need to know. PPLO is the other name. So what is the name of the agar going to be? The name of the agar is also PPLO. Name of organism is also PPLO. Name of the agar is also PPLO. And that finally wraps up most of it. This was actually not the last slide because I always lie to you about the last slide. This is genuinely the last slide which you are going to answer. Okay, bacters. Campylobacter, Helicobacter. And very simple. Campi. Campi matlab, ma'am, the name of the culture media is also gonna be Campi only. So Campylobacter media is what is going to be used in this. Campylobacter, Skero and Budzler. Skero, Budzler, not very important, but Campylobacter, very, very important. So remember Campylobacter for Campylobacterium, and even Helicobacter can also grow in it. That is what the recent studies are showing. So ये जो दोनों bacters हैं, they can grow commonly on a couple of culture media also. I think, yeah, this was tough, certainly very tough, but we finished it. But you're not going anywhere because we have two things to do. Number one, you need to finish on this table, and you need to finish on one more table which I'm going to ask you about, the fermenters. Now quick, this is for you to answer. What if you want a confidence booster, you're going to answer this and go. We are talking about mannitol fermentation, sucrose fermentation, and two types of lactose fermentation. Mannitol fermentation के लिए mannitol salt agar, you have to tell me the color and the indicator and of course the organism. Mannitol salt agar was for Staphylococcus aureus. You will say, ma'am, color में तो simple है, हर जगह please write yellow, yellow, yellow. The color of every change is gonna be yellow. The indicators will change. Please note, for mannitol salt agar, the indicator that I told you was phenol red. Okay, let's go to sucrose fermentation. Where did I teach you sucrose? Sucrose, TCBS. TCBS, that is going to be sucrose. So mannitol salt agar mein mannitol word aayega, sucrose mein sucrose is gonna come, and this is going to be for Vibrio cholera. What color? Again yellow color, but what was the name of the media, the indicator? Bromothymol blue. Now let's come to lactose fermentation. You have Cled and you have MacConkey. You have Cled and you have MacConkey. Both of them are going to give you, the yellow color is there, but Cled ke liye what is the indicator? Again it is bromothymol blue. Things are gonna change for this MacConkey. Remember, MacConkey was a pretty pink plant. So what is the color that you're gonna get over here? I'm going to get a pink color, and I said pretty pink plant. What is the indicator over here? The indicator is going to be neutral red. This is your final chart for all the fermentations. So ultimately, all of them give a yellow color. Only MacConkey is the pretty pink plant. This settlement is done. Yes, last one, and you are free for the day, and that is culture media table that we have. Yes, now this is just a very quick, I'm not even probably going to ask you, I'm just going to read through all of them in 30 seconds, so that this is how you revise on the last day of your exam. I'm starting from here. So this was the first organism, Staphylococcus aureus. This is going to be a case of, yes, this is going to be mannitol salt agar. That is why it gave you a yellow color. Coming to the next, this was pinpoint with wide zone of hemolysis, pi wide. So this must have been a case of Streptococcus pyogenes. Coming to this, again, labelings nahi hua hai, but I can see that either this is to do with a camp test or it is to do with a reverse camp test. I'm actually not asking you, but you have to answer with me. Camp test is seen in two organisms, and those two organisms are, of course, Agalacti galatine and drinking, that is Listeria. Reverse camp test is seen in Clostridium perfringens. Coming to the next, the carom coin colonies. Carom coin colonies are seen for Streptococcus pneumoniae, Draughtsman or carom coin, whatever you want to call them. Now coming to this black, there are two blacks in front of you. One black is the, remember the bacteria, the bile esculin, the B wala bacteria, that is Enterococcus. And this is what? Potassium tellurite agar. Tellurium also gives a black color, and that is going to be for Corynebacterium diphtheria. So congratulations, your first row is done. Let's go to the next row, which no one is going to forget ever. So remember, here you have the deadly one. All of these together contribute to Bacillus anthracis, and I told you anthracis is deadly, and that deadly ghost that you had to think of. So this happens to be the Medusa head appearance. The next one is going to be frosted glass. The third one has a tail, so comet tail. The next one is the egg that is beaten, so beaten egg. Then this beautiful string of pearls that you have, and finally hanging on a tree. The ghost is hanging on a tree, so inverted fir tree appearance. Coming to this, the next one. What is this? Okay, this, let me ask you. Robertson's cooked meat broth is giving a black color. It's giving a black color over here. Is this Clostridium tetani or Clostridium perfringens? So you will say the Clostridium tetani is gonna give you a black color. Okay, next one, this round. What is this? This is swarming. We've already done the full form of swarming, and that is going to be PVCs. All these organisms, PVCs show you swarming. Settled here. Let's move to the, congratulations, second row also done. Next, going on to these. What are these three together known as? Senior NTR. If you remember, senior NTR was for Clostridium perfringens. So here you have the stormy clot appearance. This is the Nagler's reaction. This is the target hemolysis, and reverse camp test I have already told you earlier. So senior NTR is settled. Agla do kya hai? These are your lactose fermenters. The pink one means that is MacConkey, the pretty pink plant, and the yellow one means that is going to be Cled. So what are the lactose fermenters? E. coli, Klebsiella. But what is this one? Is this E. coli or is this Klebsiella? Because I can see that string coming out. So this one must have been the K capsule wala Klebsiella. Let's move on to the next one, which is this blacklisted brother who has created a fight between two sisters. the Salmonella Shigella sisters के बीच में fight. So this blacklisted brother is Wilson and Blair Media. Whom will he win as a trophy? Has he got a positive reaction with Salmonella typhi or with Shigella? So positivity is with, trophy is with typhi. It's with Salmonella typhi. Coming to another yellow. So this is TCBS. Which fermentation? Common sense. Sucrose fermentation. कोई भी fermentation होगा, कौन सा color आएगा? It's gonna give you a yellow color. Settled. ठीक है. Finally, the last slide that you have, almost the last row. Which pigment red blue pyocyanin होता है? So that is going to be the Pseudomonas. Living in our country, I don't want to repeat this. LJ media is for TB. For Mycobacterium lepra, you don't have any culture media. It is 9-banded armadillo. Coming to this satellite thing, the FFF satellite, fever, influenza, Haemophilus influenza. This drop like cough droplets, whooping cough wala, Bordetella. Now we come to that animal wala thing. Animal ka milk, animal, humans. What is that? That is going to be Brucella. So this is that milk ring test that you have. I can see the answers are going so much in continuity and back to back that it is amazing to see such answers. And whenever I see such answers for microbio, it gives me more happiness than pathology, because I know paths to kar hi loge. Micro is what is giving you sleepless nights. Finally, coming to the last one, the easiest one. This is the Dinger's ring. So the ring has to come in Leptospira, Dinger's ring. And finally, these last two ones, though no one wants to answer, it is the fried egg appearance, and that is what you do for Mycoplasma. So this finally is the summary of, although this is what I showed you yesterday, so you must have thought that ma'am is gonna teach this in like 10 minutes, but it turned out to be a 1-hour class. But I hope it's been a fruitful class. But this, guys, will vanish within a day. I can guarantee. Jitni bhi achhi class thi na, it will vanish within a day from your mind if you don't sit and revise this. So what I'm gonna do, I'm gonna arrange this PDF in the next 10-15 minutes properly, and I'm gonna forward it on Telegram. Only thing that you need to do is within the next 24 hours, either you with the video or without the video, go through this once again for it to remain in your mind for a longer time. Also, this part is done. I will also be asking you randomly anywhere on Telegram and Instagram. I'll be putting up questions related to this, so that time and again our revision is also on point. Yeah, so quite a power-packed session. So I hope it has been useful, and this is gonna stay for your revision on the YouTube channel. So whenever you feel that the scare is coming back in your life, just play this on a 2x speed and revise it as many number of times as possible, right? Also, we will have another set of classes in the next week. 2 to 3 classes in the next week also. I'll update you about the timings. Many of you had requested that general pathology should be covered more because INI CET is coming. So yes, we will take up that. In the next week classes, we'll take up more of general path and general micro. I mean the topics which are asked more in the exams. Okay? Perfect. So thank you so much. Next class timings and date I'll update very soon. Tomorrow there is no class. For those who are asking what to study for tomorrow there's no class. I'll plan the classes for the next week and I'll give you information on that. Okay? Thank you so much, guys, for joining in, and thank you for being so patient on this. Thank you.